Welcome to our presentation. I'm Dr. SK, and I'll be guiding you through today's session. Let's get started and make this session both informative and engaging. Let's take a closer look at the SEM analysis of the nanomaterial. The scanning electron microscope, or SEM, is a powerful tool that allows us to visualize the surface morphology of these tiny structures at a very high resolution. When we examine the nanomaterial under SEM, we can observe details like particle size, shape, and surface features that are critical for understanding its properties and potential applications. Let's take a closer look at the history behind this remarkable invention. M.V. Arden was a German researcher and inventor, and he holds around 600 patents, which is quite impressive. He is best known for inventing the electron microscope back in 1937. This invention was a huge breakthrough in microscopy, allowing us to see things at a much finer scale than ever before. After Arden's work, researchers like M.C. Mullen and Max Knoll continued to develop and refine the technology. Their contributions helped push the boundaries even further. Then, in the 1950s and early 1960s, groups at Cambridge University took a deep interest in this technology, working intensively to improve. Let's take a closer look at the table on this slide. Here, we're comparing two key types of electron microscopy: SEM, which stands for scanning electron microscopy, and TEM, transmission electron microscopy. Both of these techniques use an electron beam as their radiation source, which is a fundamental difference from optical microscopy. Starting with SEM, it offers a lateral resolution ranging from about 10 to 100 nanometers, and a magnification capacity from 10 up to 500,000 times. This makes SEM ideal for observing surface structures and getting a detailed three-dimensional view of samples. On the other hand, TEM provides an even higher resolution. So why are we using this type of electron microscopy? Well, the main reason is that it provides the highest level of resolution available. When we talk about an image produced of a specimen using a beam of electrons, we're essentially using a much more powerful tool than traditional light microscopes. Electrons have a significantly shorter wavelength compared to visible light. This is a crucial point because shorter wavelengths allow us to see much finer details. Let's take a closer look at how the electron beam interacts with the sample. When the electron beam hits the material, it produces emitted X-rays, which are crucial for elemental analysis. We also observe backscattered electrons, which bounce back from the sample surface and help us understand the composition and topography. Secondary electrons are generated from the surface and are vital for high-resolution imaging. Let's talk about SEM, or scanning electron microscopy. It's a fascinating technique because it creates detailed images of a sample's surface by scanning it with a focused beam of electrons. When these electrons interact with the atoms in the sample, they produce various signals. These signals are incredibly informative, as they tell us about both the surface topography, that is, the shape and texture, and the composition, meaning what materials make up the sample. There are three main signals that we detect in SEM. The first is secondary electrons, which are emitted when the incident electrons hit the sample and cause other electrons to be ejected. These give us high-resolution images of surface features. The second is backscattered electrons, which are reflected back from the sample, and these can tell us about the composition because heavier elements tend to backscatter electrons more efficiently. The third type is emitted X-rays, which result from interactions that can reveal the elemental makeup of the surface. Understanding these signals is key, because they form the foundation of how SEM provides detailed surface information, making it an invaluable tool in materials science, biology, and many other fields. There are a few key points to remember to ensure optimal results and safety. First, SEM operates in a low vacuum environment, typically between 0.1 and 0.0001 pascals. This means we need to be mindful of the vacuum conditions during sample preparation and analysis. For volatile samples, we use a variable pressure SEM, or VPSEM, which can handle pressures up to 4 kilopascals, allowing us to analyze samples that might otherwise outgas or be damaged in a traditional SEM. When it comes to the accelerating voltage, For most sample analyses, we usually use a maximum of 15 to 30 keV. But if we're working with low-voltage SEM or LV-SEM, then 1 to 5 keV is typically sufficient to achieve high-resolution images. All right, let's talk about preparing biological specimens for examination. First, it's crucial that the specimen is completely dry because the chamber operates under high vacuum conditions. If there's residual moisture, it could cause issues or even damage the equipment. When it comes to different types of samples, such as wood, bone, feathers, dried insects, or eggshells, these can generally be examined directly after some chemical treatment to enhance their stability. However, for more delicate samples like tissues, living cells, or soft-bodied organisms, we need to do more. These require chemical fixation to preserve and stabilize their structure before analysis. Fixation involves treating the sample with chemicals like glutaraldehyde or formaldehyde, which cross-link proteins and other cellular components. Let's take a closer look at the working procedure. First, this slide outlines the key points that are essential for understanding how the process functions overall. Overall, this resource ties everything together by providing a practical, accessible way to deepen your understanding of scanning electron microscopy. I encourage you to explore it after the presentation and see how these concepts come to life through this virtual experience.